Okay, we have Stas, Take You, Liam, Emma, Will. All right, let's start. And Monica's here too. Okay. All right, Stas, you're on first. Updates.
Okay, so I guess first and foremost, I made those changes that based on your advice from the last meeting to the paper.
Yeah.
So maybe if you could look at it one more time and then just give me the green light and I'll submit it to JCOPO.
Yeah, give me the link. Let me just scroll down one more time.
Yep. 1 second. Link. Okay.
Okay. You change is call this reference rather than source. So you change the discussion.
So I changed figures 4 and 5, combined them into one. I
Mhm.
talked a little bit about neoantigen, the neoantigen load and how it's correlated with TMB, but doesn't really differ between the groups. I made a few changes to the abstract, and then in the discussion, I added a couple paragraphs. So
So
these
there
last two.
are some sentences that are colloquial, like with some groups, it gives us confidence that the relations observed are representative. So you can, rephrase this sentence so that it's the large sample size afforded to us by TCGA. Just the wording, like confidence afforded. I would just put it very simply, plainly, and
Okay.
more scientifically, like the strength of our work lies in, I would say large. So for some, large is 10,000.
Right.
I would say relatively large sample size patients. So if you sum it up, I guess the squamous and adeno all together is about
700. It's
the-
about 740. Let me see.
yeah. So
Uh,
like hundreds patients is like really vague. So the relatively large sample size.
I guess almost 1,000 if you include the later stage folks. But yeah.
Right. Like well, the primary strength of our work is the relatively large size of samples lies in relatively large size of samples close to 1,000 from TCGA database or something. And you don't have to say gives us confidence or something. You can take out the sentence. Allows us to stratify using multiple variables without. I mean, this is sort of like, however our work does. Introducing, so this is sort of like a no-brainer.
Yeah.
Like large sample size enables us to do that, and you don't have to write it out to say that. So I don't think we need this sentence as well.
Okay. The whole thing.
Yeah. Yeah, because like without that, the
Yeah.
paper, makes sense. I mean, just like
Right.
say relatively large sample size, that's it. But I guess then you go to limitation. That's like a one sentence is too.
Right.
So, so large sample size, and then that you were able to, I guess so I guess the large sample size, this pertains to any paper that use TCGA. So this is like, does not make our paper special. So I would say what makes our paper special is that we are the first, like, I guess we would have to go to our concept, how novel that is. So this is impact of ITH. So strength of our paper. The strength is that we analyzed ITH.
And I mean, I guess really it's the fact that we had multiple biomarkers, ITH, TMB, and then the expression data, which again, kind of going along with your point, anyone using TCGA would have that, but we kind of use it all together. Some of the older papers use a smaller number of TCGA patients just because there weren't as many at the time. But most of the ones in the last 3 or 4 years all use kind of the large groups.
So squamous adeno.
Also, most groups I've noticed don't really split the two data sets. They kind of keep them as one.
Okay.
As one non. So like we're stratifying by histology, by ITH, by TMB, and by stage. So we kind of have this complex, more complex matrix.
Okay, so I guess multiple parameters, TMB, ITH, and other expression signatures, we looked at them all together in early stage.
Yeah.
In an unbiased way. And then I guess this is the first research to show that adeno versus squamous has an inverse trend in terms of its the impact of ITH on survival. I guess methodologically, multiple biomarkers looking at it at the same time with an angle of with the differential, I guess as looking adenosquamous as a two different disease entity.
Yeah.
Just trying to figure out what the best way to frame that is. So I would put that as the first strength, that multiple biomarker.
Not talk about sample size.
No, talk about it as a number
Okay.
two.
Okay.
Um,
I added a sentence here, but it again kind of sounds redundant just because it's like we were saying, large sample size will clearly lead to less sample bias, but I did emphasize the several parameters, excuse me.
yeah, this means this allowed us, this means big sample size. So the big sample size is not what we're focused on. We're focusing on
Right.
that we're doing the multi biomarker.
What if we no.
So again, sample size is number two. Number one is the multi biomarker. Let's do let's do this. Number one, multi biomarker. Number two is the fact that we treated adeno and squamous differently. And number three is the large sample size.
Okay.
So let's just figure out some.
I'll do that during the meeting or like in the next few minutes and then look at it.
So who's the author? You and anyone helped you?
No, I think it was just me and you that worked on this.
Okay. I think you can get, maybe Haesung can help you. So there's a lot of colloquial language here.
Right.
So Haesung, if you can, Haesung, you've seen what language we use, like Dart papers.
Right, yeah.
So just work with the English grammar and just put your name in the acknowledgement for manuscript revision. We acknowledge Haesung.
I mean, yeah, I appreciate any help to kind of make this flow better and sound more appropriate.
Yeah.
As well as
Right.
anyone who wants to read through this and tell me if there's anything that doesn't make sense or is unclear.
Yeah.
This will be helpful.
So yeah, just help us with the English, Haesung.
English is my second language too, so.
Oh, is it really?
Opens the door for future investigation into interventions that can take advantage of these differences, leading to more precise and effective. So again, this is very vague, open door, what are we investigating? Future investigation into interventions. What are
I
interventions?
can be more specific. I can say immunotherapy that takes advantage of and then I can specifically talk about these enriched cell types.
Yeah. So what I'm trying to say is what we find, we have not made any connection to like a therapeutic implications. And maybe we should not, because this is so hypothesis generating. Like we're not even sure. This is one data. We haven't shown that in other lung cancer data yet. We don't know this is adenosquamous thing in head and neck or bladder.
Yeah.
So we have many questions. So I would rather say we I think we should look into the difference between adenosquamous in other histologies such as head and neck cancer, bladder cancer, esophageal cancer, where there's like adeno and squamous. And
Okay.
we could see if the same association can be found. And number two, if that is found, how can we should look into how ITH affects the immunotherapy response in those histologies where there's coexisting adeno and squamous histology. So further studies, translational as well as clinical studies are warranted to explore this hypothesis. That's what I would say.
Okay.
So what I'm saying is, anytime you write sentence, you have to be very specific, where and what kind of intervention, and all that. I know what you mean. Because we all know
Right.
the topic, but people who does not know the topic,
Okay, I'll be more specific.
yeah. So it's not just about the English and but it's more of clarity of idea.
Yeah.
So I think the more papers you write, you'll get better at that. But I
Right.
just feel like right now a lot of things are more. Yeah, I mean, this is only improved by reading more scientific papers and just like really looking into what language and expression they used and just try to mimic it.
I think
So
I think what I struggle with when writing this is that kind of the bias that I have, like you're saying, like I'm familiar with the work, so
uh,
I can say something and to me it doesn't seem vague, but
right, people who
Yeah.
does not know the topic, like what intervention are you talking about?
Yeah.
So yeah. So rewrite this and Haesung help him with the English and some expressions, like the vague expressions, you can make that more clear. I think you've gone through enough training to sort of think like me.
That's an
Uh
overstatement, but yeah, I'll do
yeah,
that.
yeah. So helps us. And if you feel like you spend enough time or comment on this graph and whatever, if you feel like you help more than just proofreading, talk to me and maybe you qualify as a co-author too. So it depends on how much time you spend on this. Just let me know, but definitely help him improve his writing, and then just submit. Okay, and you don't have to wait for my another approval. I think the main idea is there. It's just making the sentences read better. So good.
And I'm sure the reviewers will point something out that needs to be clarified, fixed, or added, so.
Yeah, what journal are you?
JCOPO was the one you suggested. I reached
Did
out
you-
to them. Yeah, go
Did
ahead.
you is it in the word limit? Did you?
Yep. It's this last I checked, it was around like 2,400, 2,500, and the word limit is 3,000.
Yeah.
So we're
And
all
you know
set.
how to make the figures, all the files and the figure legend and everything. You've done it before, right?
Yeah.
So yeah. Okay.
And then a few months ago, I reached out to them for kind of a pre-submission screening, and they gave me the green light to submit. So I'm all set.
All right, then let's do it. Okay. And make
Um,
sure after you submit, check it almost like weekly basis, because they will return it like, oh, fix this figure, like
Oh,
fix
okay.
this, and they return it right away. And if you don't check it, and
Right.
I know I'm the corresponding author, but these emails go to sometimes like spam folders because they're auto-generated emails, right? So
Yeah.
it's hard to pick those emails. So it's better to go into the author login toolbox and then just check it every few days. So literally.
All right. So then I'll make those changes today, and then I'll just talk to Hassan about
Yeah.
the time frame for edits, and then go
Yeah,
from there.
Haesung, just help him submit. You have access to my ID, password, and JCOPO. Okay. All right.
So I would be submitting through your account. Is that what you mean? Or
No, you submit,
Right,
but I would have to endorse it.
okay,
But that Haesung can help.
okay,
Yeah,
sounds good.
because she has my account to endorse it.
Right.
Okay, any progress on the other data, the projects?
Yeah, so last week Hasung shared a list of about 200 patients, a little over 200 patients from, I guess this is mostly from the radiomics project. I went through this list and about 60, a little over 60 patients, 61 patients had Tempus IDs or accession numbers through based on the Tempus portal. So you put their information in and then it'll spit out an ID. So about 60 of the 200 had a Tempus ID.
Hmm.
Of the 60 who had Tempus IDs, 53 actually had data in the S3 bucket for Tempus. So there were about 8 that had IDs, but we just don't have access to that data, or it's in a different bucket.
Hmm.
I then took the DNA and copy number data and ran it through PyClone and generated ITH. So now we have ITH for 53 of those patients.
Oh. Okay.
And then the other thing we talked about with, or I talked about with Hasung, is the fact that these ITH numbers are kind of based on a really simple set of parameters. Any cluster with 2 or more mutations is considered a clone. We can discuss what kind of rule set we want to set up for what is considered a clone, sizes, presence of specific mutations, and kind of going from there, defining what a subclone is if we want, and kind of going. So that's kind of the next step, as well as figuring out where these other eight patients, where their data is.
Mm-hmm. Hmm. Hmm. So that 200, so Liam, that 200 patient is the ultimate radiomics patient that we gathered?
Yes. So the ultimate number of patients we have on that list is 209. So I think that's what he is referring to. We're still waiting for like 60 or so of the CT scan images, which is we submitted the application for, and we're just waiting for them to give us the data. But this is the Tempus side of things. I think Dr. Hassan can speak more into it.
Yeah, it's total of 209. We uploaded the CDs for 138 patients, and now we have requested 60 plus CD files, and so we're trying to get all the CD files for all the 209 patients.
Okay. Okay. 53 is kind of small to, yeah, really like, I mean, it's doable. So yeah, look for that A patient where they are, the data. And then I think, with 53 data, play with it. And maybe, you can do Zoom meetings with Haesung. This will be a nice opportunity for Haesung to learn, how do you assess clonality. Just learn from, so that you guys can discuss and come up with brainstorm, come up with better ideas and present that in next meeting. So the simple question is, now we have the patient outcome data and we have ITH. We can measure it in many different ways as we want, and then we can, combine it with TMB, PD-L1. So PD-1 TMB is like the known biomarkers that are not perfect, and a lot of them I would say got chemo-immuno combo. So if you get chemo-immuno combo, TMB does not mean much. And PD-L1 doesn't mean much. Okay, but we do know ITH it's important whether you get targeted therapy, immunotherapy, or chemotherapy. So maybe show the superiority of ITH versus PDL1 or TMB, or additive or added value of utilizing that. We have VeriStrat that showed it's a proteomic signature from serum that showed that it works even if there's a chemoimmunotherapy. So with that, I think you can look into many different biomarkers that you have and see how that separates the curve, sort of PFS or OS. But the goal is, what is the best? So start with ITH because it's an ITH project. See if ITH separates the curve nicely, and and then methodology wise, try any method that has been described by different groups, including including what you did with that Adams Squamous paper, but use different cutoffs and kind of see what cutoffs makes these curves separate better. So work with the data and kind of show us what you find. So let's start there. We want to do the exact same thing with the Guardant raw data. And eventually we want to combine the Guardant and Tempus data and show there's a utility in utilizing both. Would have been better if we had several hundreds, but we start where we can.
Well, one thing to note is that these patients are stage 3 unresectable patients or stage 4 who received IO as frontline therapy. But what you said last time was to, collect data regardless of their line of therapy. So, I mean, right now, maybe we can start with these 53 plus patients, but in the future, we can add more patients who received it as second, third, fourth.
Yes.
And then do like a neoadjuvant study in the future as well.
Yes. So we don't, I think this 200 is only frontline. Good point. So we do want, so that's an older data, but we want to look at the second line immunotherapy too. But that's when immunotherapy was not approved frontline, and that's kind of a couple of years ago, going back. And then we are now using immunotherapy as a neoadjuvant therapy. So that's another different group. So we start with frontline immunotherapy, but then like Haesung said, we need to now get data on neoadjuvant group as well as the second and beyond immunotherapy group. But the caveat is, if let's say I use the immunotherapy frontline, then I don't care what immunotherapy they got second, third line. So these are not, they are counted as a frontline immunotherapy. So what matters is when did they start immunotherapy? It's a second, third. The first line is just carbotaxol, and the second line is Pembro. Then it counts as second or beyond immunotherapy group. And it used to be that way because immunotherapy was never approved as a first line therapy in lung cancer, let's say 3 years ago. But the data from like 5 years ago to 3 years ago is like immunotherapy is all second line, and all these like a beautiful paper that came out, Science, Cell, like all these papers, Swanton papers, who are using immunotherapy as a second line. So it's an old paper. So anyway, we need 3 different cohort, as I said. So Haesung and Liam, you guys can be in charge of sort of what would be the most efficient way to get that data. Second line, beyond, and the neoadjuvant data. So like a build database, and it can be used for any projects, like proteomic project, Tempus project, a Guardant project, and I take you a CHIP project. CHIP is another area that we are looking into. It's called clonal poiesis of insignificant potential, and it's, you could call it pre-MDS, and the effect of that in patient treatment outcome is something we could look into. So we'll talk more about it. My point is, we should get those clinical outcomes. But start with 53. Work with Stas, Hesong and Stas, and let me know what you guys find. Okay. Guardant raw data, where are we with that?
So I,
Liam
yeah,
or
go ahead.
Stas? Yeah.
Well, I after our meeting last week, I reached out to Leslie B,
Yeah.
and it sounded like either her team or they met with you guys, and it was just a very strange communication. Anyway, so it was unclear on my end whether there was communication between her and her team with you guys.
So Liam, just follow up with them and
Yeah, so
gave the protocol. You gave the protocol, right?
yes, I sent them the protocol. Leslie actually just sent an email like 30 minutes ago saying, sharing an update. The draft DUA is with their legal team, the Guardant legal team, for review prior to her sharing the data to us. She says she will send it over as soon as she has it back from her legal team. So it's in the works. It's in the works.
Okay. So once they send data, whether by cloud, share it with Hesong and Stas, and Stas, I think we said so we did say that we want the raw data, but then if you can't, the copy number and the allele frequency is all we like minimum data we need for all the genes that were sequenced, like 500 plus is what we requested, and they said they have it. So we're really hoping that they would give us that data. So yeah, if you get it, do some, play with it, see what you find, and compared to what you have with Tempest, is it something that can be mergeable? I think, what's different.
Yeah.
Yeah, let us know in the next meeting.
The other, well, once I get the data, I can work with it. The other thing that Tempest has done for us before, not Tempest, sorry, Guardant has done for us before, that may be a workaround if they can't give us raw data, is if they can prepare the input for PyClone. This is something they've done before when we first talked to them. So that's another option if they're unable to kind of give us the raw data.
Yeah, let's see what they give us and then go from there. But yeah, good point.
So
yeah,
just as far as kind of like the action items going forward, I think Hesong and I guess Liam and I should meet this week and figure out what relationships, or maybe just Hesong and I, what relationships we want to look at, how we want to, what definitions, kind of more of the actual comparisons we want to make.
yeah.
So we'll do that.
All right. Anything else?
One other thing. So as I mentioned last meeting, I'm doing like a medical informatics elective with and the director for that elective is a guy named David Leibowitz, who's very active in the Epic data side of things. He's a clinician at Northwestern, but this is also part of his appointment. And he mentioned to me that there are a few tools that can let you search the Epic databases a lot more kind of programmatically and a lot more quickly than just looking at charts. Specifically a tool called Slicer Dicer, which it sounds like some clinicians have access to. And he said that anyone can become a quote unquote super user. What that really means is you have access to some of those kind of backend databases, which again lets you parse patients, see who ordered what, maybe even do some chart parsing. And he says that access to that tool right now is limited to folks who want to do quality improvement projects. So I just wanted to let you know, if you have a quality improvement project, you can, or ideas for a quality improvement project, for example, which patients are getting Tempest or Guardant tests, and how that affects the quality of their care. We might be able to get more information about just how many patients of yours have that where it's kind of just, you're not supposed to use that tool for research, but at the very least, it'll give us a baseline to work off of.
But what
Um,
is that tool used for then? If not for
quality, it's primarily used for quality improvement. So looking to see which patients are getting what, when, how, and then ultimately how that affects their care or the cost of their care or the efficiency of your clinic, you name it.
So before we apply or do anything about it, why don't you learn more about that
Yeah,
program and what you can actually do and how we can help our research? And like if you kind of play with it and then find it useful and not so useful, then you can let us know next
okay.
meeting.
Yeah, that sounds good. Yeah, I just wanted to check in with you before I talk to him again about it.
Sure. What about EDW? Are you not involved in
That's
that?
also part of, that's kind of like, it's, I think Slicer Dicer sits like on top of EDW, or there's some relationship, but it's it's a more simpler tool to use. That's also part of being kind of a super user. So
Yeah.
that's another thing I'll talk to him about and see what really the criteria for a quality improvement project are to gain access to these tools. So I'll get more information, but I just wanted to check in with you before I did any of that.
Sure. And Liam, maybe you can add Hesong and Stas to our retrospective lung protocol, because that will cover the whole Tempest project, and garden project, everything.
I believe Stas is already in there, and I'll check to see if we can add Dr. Hesong.
Yeah, yeah. Okay. Okay, yeah. So learn about EDW as well. So maybe you
Yeah.
could, for these projects, we have someone called Ben helping us, but like it's good if you can query data yourself.
Yeah.
Yeah, I know I can, but I just don't have bandwidth to do it myself. So yeah, learn about EDW while you're in that medical informatics program. That's sort of the kind of basic of
Yeah.
the clinical data inquiry. So
And
good,
and
good.
then my only last update is that as part of that elective, I have to do like a project, and the project I mentioned to you last week is looking at tumor heterogeneity on whole slide images. And so I've kind of got the ball rolling on that. I've been able to train some models. They're not performing that great, but I'm kind of just learning the tools first, and then I'm going to start. And then once I have any kind of like real findings, I'll present them.
Yeah, present them. Let us know what you find. I mean, that's always something that, we wanted to do. So any heterogeneity index you can come up with by imaging analysis,
Yeah.
let us know your rationale and what others have done
Yeah.
in the same field, and talk to Lee Cooper, because
Yeah, that's
I want you to get his input as well, and see what he says about it. And then whatever you find, because we have scanned our own patients treated here, we have the H&E slides, then you can analyze that, and we have clinical annotation of, outcome. So we can see if any of that, image heterogeneity translates into differential in prognosis in
Yeah.
patients getting immunotherapy. So yeah, that's very exciting.
At this point, I'm looking at just simply the relationship between, I know which slides have high pi clone and which slides have low pi clone, and I'm really just trying to see if the image tools can spot a difference, and then hopefully create an independent heterogeneity image-based score. But I'm just kind of at this point just learning how to use these tools and getting familiar with it. But
Right.
there's some work out there about it, so it's pretty cool.
Yeah. So yeah. So here's the idea, and Hesong and Liam, you guys can, be involved in this. So the idea is just looking at H&E slide, tumor and stroma, and looking at that. And I think there's heterogeneity. Like if you look at adeno and squamous and mix them, there's so much heterogeneity. So you want to limit other source of heterogeneity as much as possible. You can't combine lung cancer and small cell lung cancer and non-small cell together, because there's so it has to be same diagnosis, and same subtype, and then within that, looking at heterogeneity by the morphology, does that have any correlation with the clonality? So, and showing that correlation is actually first. Yeah,
Most
yeah.
of most of the work I've seen so far on this, kind of the most similar to this, they'll cut up the whole slide into tiles, and then they'll only pick the tiles that have more than 50% nuclei, and then they'll look at the morphology, shape, you name it, a ton of features of the nuclei, and then based on that, they'll calculate some sort of heterogeneity score. Because it's hard to see the actual cell membrane, but nuclei are very easy to filter and spot and qualify or characterize. So,
Good, good. All right, keep us updated. Very fascinating project. Okay, let's so Emma, it's been a while.
Hi. Yeah. How are you?
Good, good. So yeah, let's get your update. Tell us what you've been working on.
Yeah, I'll send some documents over in the chat. The first one is the draft of the variant classification project, and the second one is like the chart containing all of our analysis. The draft is pretty similar to the versions that you've seen before. We haven't worked that much on it, but the table basically we just completed. And if you look at the table, you're gonna see a lot of like very statistically meaningless values. And that's because an issue that we've been running into in our project is the low sample size and also an imbalance between like the benign and pathogenic data points. So we calculate our performance metrics. It's like coming up with like undefined or 100% because there was like maybe just one pathogenic variant for that specific analysis. So I guess what we've been mainly working on in the spring was since our original analysis was like calculating those metrics overall, like with all the data points and then stratifying the data points with either cancer type or gene type. When we did the stratification, like that's when we weren't seeing that much meaningful values because there's just not a lot of data points. And there's an even bigger imbalance in the two classes when we stratify. So we were trying to like brainstorm other analysis that we can do since we didn't think just doing the overall analysis would be like enough for a manuscript. So some ideas we had were combining like making combinations of tools, individual tools to see if they perform better than like individual tools. But at the same time, I don't know if that'll be meaningful because some individual tools in itself are already stacked tools of other individual tools made by the developers. So yeah, and another thing we could do is like the developers of each tool release like their own publications with like metrics, performance metrics using their own data. So we were thinking maybe we can compare our results with their results to see if it matches, and if it doesn't, like why it didn't. Or like each model has parameter settings, and we've been just using the default settings that the authors or the developers recommended, but they are able to be like tweaked. So maybe we could tune it to see if changing the parameters will increase performance. But yeah, we've been just like throwing these ideas around, but I think we just have to like sit down and pick which one we actually want to do and which one's like plausible. So where we're at right now, Sally, Eileen, and I are meeting today. I think we'll probably just like solidify the introduction and methods like this week, because Dr. Easer, like she doesn't know her schedule for ICU yet, so she's not able to meet with us. But when she gets her schedule, we'll probably meet with her and decide which additional analysis we'll do and how to write up and like talk about how to write up the results and discussion. And we're hoping to be done by the end of August, but I'm guessing we'll have a pretty good draft by like first or second week of August.
Okay. So this is is this the very first one?
this-
The second one, right?
Yeah, after Eugene's.
Because Eugene and Sam worked on it, Samantha.
Yeah.
Do you know where that paper is?
The last time I talked to Eugene, this was like months ago, but I remember he said he submitted it to multiple places, but it didn't get accepted. So he was just kind of like not sure what to do.
So why don't you carry that paper as well? Because he doesn't know what to do. So the answer is submit to the next journal
Okay.
until you find the home. And so here's because that's the first paper that
Yeah.
has to come out. And then you have the lung cancer paper and then solid cancer paper. And then we have potential, these are validated targets, right?
Right.
Actionable targets. So the history, so for those of you who do not know what this project is about, I'll tell you that we see mutations in the NGS report, and then sometimes we're not sure whether it's actionable or not. We're not sure. And we rely on the company who made that report. And the whole idea is really being critical and kind of see what is the current performance of all the algorithms out there that says a certain variant is functional, actionable or not. Okay, so we first start with known actionable targets and see whether the current algorithms perform up to our expectation or not. Very simple. And we're gonna say some do well, some don't. And the more you combine, like Revel, or maybe it does better. So I think, we don't need a deep statistical exercise here, but whatever is standard, how do you compare accuracy of certain assays or algorithm output, and what is your gold standard? And you compare it and say, this is what we see. And then it's just like reporting the simple concordance. So let's tackle your question one by one. So I don't like, number one, just looking at the standard value approach suggested by each algorithm is fine. I don't want to go in depth on that. Next is combining different tools ourselves and coming up with a different, that's too much. I think we can just stop at evaluating the performance of each tool. That's okay. And the Revel is the one that kind of combined everything itself and scored it. So this is sort of like a mixed platform. And we can emphasize that, but so in my mind, if they reject it, they'll reject based on the significance. Like reviewers say, well, this doesn't really have clinical utility, or has enough impact to change research or clinical practice, then they'll reject it. If they have a problem with, oh, you should not have calculated this is wrong way to calculate it, or specificities, you should not combine these, or they have any methodological suggestions, we fix it. If they have a difference in opinion about its impact in the field of research, then we go to the next journal. We just have to find the right reviewer. But at least methodological suggestion wise, have you run across suggestions or brainstormed ideas that you think we need to fix, Emma?
Can you repeat that? Like,
So any suggestions in the method that so I gave you like the overview, but
Mhm.
like anything you want to fix. I know we have to, I mean, writing discussion and, you know, making tables and figures are different issues, but the whole research structure, like, should we fix anything or add something new to make this more presentable and more, I would say, applicable to the real world?
I think, in my opinion, like our biggest issue is just the state of our data, just
Mhm.
the lack of data and the imbalance in data. Because if you look at Eugene's paper,
Mhm.
he also doesn't have that big of a sample size, but at least the number of benign variants and pathogenic variants are pretty equal. For us,
Mhm.
not. And it gets even worse when we do other analysis. So
And what was the reason for that?
I think when I reread his paper, he collected his data a little differently than ours. I'm not sure if I agree with his method either. I'm not sure why he did it that way. But basically, what we did was there was like ClinVar and like OncoKB, like all those variant databases.
Huh?
We just looked through all possible variants in those databases, like within our selected cancer and gene
Genes?
types,
Yeah.
and just pick them out. And if
What was
they
the
were,
criteria of picking them?
it was like if, so ClinVar had their own um, like pathogenicity kind of thing, and then like OncoKB had their own. And then we also use like My Cancer Genome, I think. And if it matched like 2 out of 3, then we included it in our data. I'm pretty sure. I might have to look into that again.
Yeah, I
Uh,
think that's what you did for picking out pathogenic genes.
Yeah, we also did that for benign, I'm pretty sure. But what Eugene did was he like picked out, I think, benign variants from ClinVar only, and then like pathogenic variants from other databases, because he was saying like ClinVar didn't have a lot of pathogenic variants. Yeah. But I don't know how, if that's a better way of collecting that or not.
Huh. So there's, we say gold standard, but it's like the gold standard would be you have a like a mouse model or a patient data. That says, the drug worked. The
Mhm.
tumor in mice or human shrink by inhibiting that specific mutation. Mutated clone with the targeted therapy. But here, so I guess so my point is there's no gold standard. There's no, I think the reviewer is gonna say, oh, the way you did it makes sense. Maybe you did it don't make sense, and it's still very subjective. But I agree that the numbers, if it's equal, it's better. But if you explain why the number is the way it is because of our criteria, because we define pathogenicity as, find these variants according to this criteria, you can say that in your methods, or you could sort of like do a flow chart, like a figure with the arrows and boxes, and put that in the supplemental figure, so that people know how you derive these numbers, pathogens and pathogenic variants and benign variants. So my answer is, I think it's too much to rerun everything by changing this criteria at this time. And it's better it's equal, but if it's not equal, I think we can still analyze the performance. If a reviewer has major problem, the numbers are small, it's not equal, I think it's statistically weak. We understand it's weak, but this is with that criteria, best we can do, and move on to the next journal. I don't want you to just to get this into a journal, that journal, it's too much of a work. So as
Okay.
long as we justify ourselves in Eugene's paper and your paper, what is different, and it may be different paper by paper, it doesn't have to be exactly same. And it's just how we define it and just explaining why we did it that way.
Okay.
Yeah.
Then
Next.
I'll relay that to Dr. User and we'll probably just start writing results and discussion.
Right. And talk to Eugene and get his paper and get that submitted on his behalf.
Okay.
So is
Oh.
Dr. Chiu still involved?
He's not. No, it's mostly Dr.
Also-
User.
When was he last? Like, when did he last talk to you?
It was honestly at the AACR 2022.
Uh-huh.
Yeah. When we submitted the abstract, but that was about it.
Hmm. So who's been like the main person writing this up when doing the analysis? You, right?
So it's me, Eileen, Sally's also working on it. And then Dr. Iser and Dr. Yoon, he's just been coming to our meetings.
Hmm.
To clarify, Emma, though, so Sally and Dr. Uni's working on their version, not this particular draft, right?
So I'm not sure because I was confused about that too, since we were also supposed to be working on like Grace's project, but we've been just like talking about it as a whole, not really like dividing it up. So I'm not really sure, like, I'm gonna have to ask Sally if she's been working on Grace's project, but I'm not sure.
Grace's project, as in,
The one that's not FDA approved yet.
Yeah, the potential target.
oh, is that the first one or
That's the third one.
that's the?
And the
Oh.
Fourth
Yeah.
one is Sally and Uni is what, yeah, fourth is the immune modulator genes, right? That's the fourth one.
Okay, so Sally and Dr. Uni is working on the fourth one. Emma, you currently have the first and second one. This is the second one you're showing us. And then Grace Lee has the third one, is what you're saying, right?
Yeah, but I think we're supposed to write that one too.
Got you.
Yeah. So as you can see, it, right, publishing something takes takes a lot, it's you need persistence. That's why we have a lot of abstracts and of only much less number of papers. So yeah, thanks for taking the lead, writing this up, but talk to Eugene and get his paper and let's publish that as well. I mean, that's written up, that's just submitting. So
Yeah, everything's
yeah,
done.
yeah, everything is done. We don't have to fix anything, but see what's fixable according to our discussion.
Okay.
And submit, and then you can talk to me and Liam about what the next journal should be. We'll
Mhm.
always give you the suggestion, but you have to get the list where he has submitted before.
Yeah.
And then I'll give you the next suggestion. Where are you submitting this one to?
We don't know yet.
Oh.
We have to talk about that too.
As well as the next, the third one is the potential targets, right?
Mhm.
And that's all solid tumor as well, right?
Yeah.
Yeah.
Okay, I'll talk to Eugene and get the list from him by next week then.
Yeah. So
Uh,
Liam, just talk to me and I will give you the list of all the potential target journals
Okay.
today. And just yeah, let's do that today.
And Emma, so do you have Eugene's paper, like the draft, or do you have to get it from him?
I might have to get it from him. I'm not sure where it is.
Okay, if you can also get that from him. And so, Dr. Chu, when we're submitting these, do you want them to be submitted in order, or can we submit them simultaneously?
Simultaneously in different journals, as you know. But the order is in, like we said, first, second, third, fourth.
Right.
That order is preferred.
Mhm.
We should cite each other because these all have posters. So you can cite others if needed, the posters. But Eugene's paper kind of, we wrote it up like 2-3 years ago. So I do think it's still publishable way it is. That the only thing is the better algorithms coming out, and you know this we might be talking about something that's outdated because you know the pace of the scientific advances we see. But anyway, let's submit, and then if the other authors tell us better suggestions, then we'll fix it. But as far as the journal is concerned, I'll give you my recommendations today. And there is Dr. Jian is here. There is a program that spits out, like if you put the title and abstract in it, what are the journal that is, I think Dr. Jian, do you know that website or the program? Can you if you that you
Yeah, there are many things, but basically you can go into Elsevier and journal searching service.
Journal searching service.
Oh, find the journals. I will put this
Find journal.
URL to
Are you this one? Match my abstract. Journal finder. We can try that. Oh, this is heterogeneity. Copy. And then put the abstract in. Science journal, turn it. Human pathology. Veterinary urology. Time to first decision 1 week. Time to publication 3 weeks. Wow. Translational research. Journal of Molecular Diagnostic. It's $3,000 to publish it there. Helion. So Helion is pretty broad. So remember this, Emma. Here we are. You mean Elizabeth breasts? Mutation research. Broad scope, open access journal. Data in belief. In brief, what is that? Fully citable and index and allows you to describe your data and make it discoverable and shareable. Methods. X. Methods and protocol article. Software publications. Very interesting. It doesn't give you that. Not really impressed. So I would translational oncology, translational research. So remember this one, translational. There's a journal called Cancer too, Cancer. Okay, keep that in mind, and I will do some more research and let you guys know.
Thank you.
Other questions, Emma?
Not right now.
So you and Eileen's working together. Isol is helping you. And you guys have planned to, get Eugene's paper and the other two papers out. You're gonna help. And then once you kind of done all the things, run the author list by me. I want to be fair and credit the ones who work the most.
Mhm.
And we'll talk about the authorship once you kind of done all your work. And then I think Sally and Uni's doing the 4th projects together. But if they help you write any of those 3 projects or help you submit whatever, tell me what they did. I'm willing to acknowledge them as co-author.
Okay, I'll keep track of everything.
Being kind of one in charge. So Emma, what is your plan? I know you like data research.
Yeah.
So yeah, I know these papers will, it will be a good experience for you, but what is your plan?
Well, right now I'm just trying to work. Just trying to get a job. Yeah.
But what kind of data do you like to work on? Like, I know this is more like a genomic data, but
Well, I do like healthcare IT type of data. That's what I've mostly worked with, but yeah, I think right now I'm just exploring, but yeah.
yeah. No, as you heard, Stats and Hesong were very interested in heterogeneity. This is
Mhm,
all genomic data.
mhm.
So if you're studying data, genomic data, RNA, DNA, and then we have mass spec. Like you learn organic chem. we,
Yeah.
we need mass spec.
Yeah.
And this is all data. What is the peak level?
Right.
And how do you analyze that data? This is what I'm working with Dr. Ether.
Mhm.
And you're more than welcome to be involved in these type of more in-depth data analysis. Not just, like, more like finance data. There's, healthcare utility data, IT type of, who accessed what, what was the number, but more like, these are the data coming from each essays. And the whole idea is the points, like the peak number in the mass spec. And that's supposed to present some sort of proteins. We don't have to know what they are, but the pattern of the peak and all that, can we use those data to predict immunotherapy toxicity? Those are the projects that we are currently working on. And you're more than welcome to join any of like in-depth data analysis as you want. I'm just
Yeah.
throwing it out there.
Thank you.
You like data analysis.
Yeah, thank you.
And talk to Ether about those data she's working on. She'll let you know about what we're gonna do. And you can be involved as much as you want.
Yeah, I'll ask her about it.
Okay, good. Okay, let's move on. And Dr. Chion, can you give us update?
And so two original articles are still under review and under review for revision. I sent the email, but they're still working on.
Okay.
And the radio mix is my main project in these days. So we have 131 patients with 273 intrathoracic lesions. So we are working on the making manuscript. We started writing manuscript yesterday, last weekend. So now we try to compare between durable responder and non-responder, as well as between intrathoracic and extrathoracic lesions. And we want to see that relative changes per lesion. So, and we are waiting the Professor Urey's result. The problem is we already almost finished the introduction and method section, but we don't have research at all. So today I'm gonna test to Professor Urey, and I really want to visit him with Peter. And yeah, we'll see. So Peter will show you the introduction part. And also Monica is working with me making tables. But I want to show you this. This might be our figure 1. And I asked to Professor Urey to make this empty part regarding harmonization model. And he said yes, so we can wait. And second one, you asked.
Go
Yeah,
up.
sure.
Training and validation. So 273, what does that mean?
273 intrathoracic lesions.
Right, but the red
red one, I don't know. I have to revise it because usually in radiomic paper, they use 60 to 70% for training set and the rest of them using for validation set. But we don't know how many cases Professor Urey will use for training or validation set. So if we, when we get the result, maybe we can figure out. So that is just assumed.
Okay.
And any other feedback for this figure?
No, I think Data analysis, it's such a broad, so I guess the model, with the model, you're data analysis. So the training is, so in the training phase, you are training whether the radiomic features can, the mix or the
Mm.
algorithm can be made to
Mhm.
predict outcome. So model development and validation.
Oh, and we're looking at the mixed response versus the universal response as well. Yeah, yesterday we categorized it, and there was 43 patients who showed a mixed response.
Good.
But then, yeah, so I think we can.
Look at that. Yeah. So I think model development and validation does
Mhm.
involve, outcome association. So, I mean, it just gives you this just, I mean, if you just look at the flow and the figures, it gives us an idea that, just using the features you have model and then you just correlate that, clinically. So
Mhm.
I think what you need is at the validation development phase, you have a model that predicts outcome.
Mhm.
But then but then it gets confusing on, I guess, like you need to high orders validation. So in the data analysis, you could just say clinical outcome analysis or something. Clinical,
Wow, okay.
yeah, because it's okay. I think they'll understand.
Mhm.
Yeah, okay, you can move on.
All right. This is waterfall and swimmer plot for changes per lesion. So which one is better?
Yeah, this one.
Okay. And I want to ask you one method. When I look at the changes per lesion, actually I use the concept based on the resist. If more than 30% decreased, I define as a decrease. And more than 20% increased, I define the increased. But is that okay, or do I have to use the absolute difference between two time points?
Can you say that again? I didn't understand it.
If the lesion was 23, then it became 22. Only 1 millimeter decreased,
Right?
but it could be decreased. But it's too small number, 1 millimeter, only 1 millimeter. So
Right.
I use the concept that resist uses. More than 30% decreased could be decreased. And more than 20% increased, then it could be increased.
Oh, I see what you mean.
Is it okay?
Oh, per lesion. Oh yeah, I mean, I guess the waterfall plot is waterfall plot. You'll draw that, but
Mhm.
So yeah, you can use the same concept of PD and PR, the same per lesion. You can, but
Okay.
if your method, per lesion response is defined as PD, PR, using the same criteria,
Mhm,
and you can call that, yeah.
Okay. Okay. That was my question. Yeah. And I will let you know the further process when I get the feedback from Professor Yuri.
Yeah.
Mhm. Okay, so next one is a liquid biopsy. Usually for paper review, we need two reviewers, but then one is completed, but it seems like one reviewer still waiting, or yeah. So it is still in under review.
Oh,
I
okay.
will check, even if I go back to Korea. I can check.
Okay. Oh, hold right there. So, Liam, write down the list of these journals, because these are like the very broad, and you can write any translational papers here. And you can add translational oncology and translational research. Those two papers down here. This is for Liam. So iScience, Helium, BMC Cancer Scientific Reports, and +1. eBiomedicine is but I think it's high bar. There's yeah. So maybe so these 1, 2, 3, 4, 5 plus the translational oncology and translational research. So I don't care what order they're, to me, they're very similar. And just mix it up in a different order. And Emma and her team can submit to all these journals in different orders, so that they don't like submit all papers into same journal at the same time, but use a different sequence. So why don't you just talk to Emma that we could just do that. Okay. Yeah. Okay.
Understood.
All right, Dr. Chion.
And then my final project is ctDNA for breast cancer. And company and Korean team, they decided to add 5 more patients with recent samples. And also we will collect serial samples for 34 patients. What we did before, but the patients are still in surveillance, so we can get their samples more. So after I going back to Korea, I think I can collect those samples with the Sujam. So we will send it to company, and we will make a better paper with those research. And still we're writing methylation research and tissues.
Okay. So did you, I did ask you this before, did you find any recent publications asking similar questions?
Not publication, but in ASCO there was very similar studies. A lot. So I think we have to publish as soon as possible.
Right.
So it is kind of a competition.
Competition right now, because everyone's doing it, and
Yes.
the
Mhm.
idea people understand. So it's not like they don't, I think it's clinical if it's the first of its kind. HER2 neoadjuvant space, if no one has shown it before, has to come out. Okay.
And now because immunotherapy is available for breast cancer, so the ctDNA field will expand more.
Oh, okay.
That's all I have today for my final last lab meeting in US. Okay, let's go.
Yeah.
Just in US.
But yeah, so I think even in Korea, you should be able to just quickly come in and update, because this
Okay.
is like after your work is done, right? This time is, yeah.
Yes, it might be nighttime.
I-
on Tuesday. So yeah.
Good. Well, thank you for all you've done in our lab, and then we expect to continue to work with you.
Of course. Yeah, it was my pleasure. Thank you.
Thank you. Thank you. All right, we'll go to Dr. Hesong. Oh, before going to Hesong, Monica, I know you've worked with Dr. Jian. Can you just give me, I know this, she talked about radiomics, but can you give us update on what you actually been doing?
So since last week, I have been more working like as a help for everybody. I guess I helped Dr. Hesong had with some data collections through Epic and a bit of segmentations with Dr. Jian, and now we're working out on tables for the manuscript. So that's pretty much been a helping hand for everybody around. I hope so.
Yeah. So you will be the lead for radiomic project going forward. So I know you'll be here for a longer time. So I really want you to know, nuts and bolts, everything about the radiomics project, what programs we're using, everything, the details. And Peter knows a lot, so learn from Dr. Jian all the clinical insights, and Peter can tell you the technical aspects. And you'll be the point of contact for radiomics, and you'll carry on the projects, like literally like carry it on. So you are going to be the lead. I know Dr. Jian will still be involved and mentor you, but I want you to have the ownership of the projects, so that people who come, because the Peter and Dr. Jian will leave the lab. So if anyone has any questions about radiomics, they will go to you. And you can go to Dr. Yuri together today and meet him and build a relationship with him. He's a great mentor. I want you to learn from him directly. Okay. So it's all about how you as a person, build relationship, how you communicate. You're not waiting to be spoon-fed. Peter can tell you his experience, whether he was sitting waiting to be spoon-fed or he was actively reaching out, asking questions. So those are the qualities I look for, how proactive are you? You cannot, some people just go to lab and like, oh, this lab, I don't think I'm learning any much. Like no one's teaching me. And like some people just feel like they do some really laborious work and didn't feel like they learned anything. They leave the lab. But here I help guide you to interesting projects. And my role is guide. You guys are self-learning. The methodologies out there. People learn everything through YouTube, and there's online courses that are free Coursera courses, and you have great mentors, or the prior lab people. They can teach you whatever they learned from our previous lab members here. So there's no limitation on what you can do here. So so just like spend a few more days with them, make sure you get all your questions answered, and build relationship. All right, that's my 2 cents. Let's move on to Hesong.
Okay, I'll talk about the DART papers and then a bit about transplant and then Tempest. I guess I'll start with something that's shorter. The transplant abstract, as soon as I get that ready, I'll show that to you. And then for Tempest TAD project, we did get the data delivered to Lens, but I was somehow not able to see the full data summary and get like the analysis of like how many patients have the TMB data and blah, blah, blah. And so they said I'll have to wait a few more days to get the raw data. So I think just a bit more waiting for that one. And then I haven't heard back about the Mucinous Discover project, like regarding like sending the 2 fellows to their headquarters. I sent them an email last week. So perhaps we can wait till the end of this week and then shoot them an email again.
Yeah, I think what we are
Something like that.
learning is it takes so long, right? To like do anything. Yeah.
Yeah, but in the meantime, we have a lot of projects starting up. And I guess that it looks like I'll have to write another proposal with you, which is also very exciting. But yeah, as soon as you get the data, you can just send it over to me and then I'll get started on that as well. And then for Dart projects, I didn't realize how much time it took to get all the formatting right. And so Liam and I worked on like shortening the abstracts, and then there were very like small, minor, very many things to consider. But the biggest bottleneck right now is to shorten the references, because these are very rare rare cancers that I like reference to many like case reports, because there weren't too many like big papers that like that summarized, did like meta-analysis of everything. So yeah, I just ended up having too many references, so shortening them would be a challenge. And then we're still waiting for the risk tables for a couple of cohorts. And then I want to show you this is for the thyroid cohorts.
Wait,
It's
before
been
we get to thyroid. So
Yeah.
my suggestion is keep the longest form, because every journal will have different word limit and different reference limitations.
Mhm.
So keep the original one, and then you can always reformat to a journal that has more strict requirements.
Mhm.
Yeah. Okay.
Yeah, I was trusting on the Google Docs version history, but yeah, I think that's a good idea. I think I'll lose track of everything. It's been
Yeah.
a challenge. Yeah. And then this is the thyroid cohort. Because they also have like a 6-figure table limit, I'm trying to clump everything together into single like figure, but the problem with that is that the font sizes get way too small. This isn't obviously final, but
Hmm.
this one is okay, I think. This is font 12, which is a requirement of this journal. This
Uh-huh.
is JCO. But I think this one looks okay, putting
Yeah.
these 4 figures here.
Yeah.
But this one, which has, the full data, the correlative data, and then the Oncoplot, and then the PDL one, this one is it should be bigger than this. And then the PFS-OS, I'm not sure. The font get really small. So I was wondering if you might be interested in taking something else.
I see. So figure A can be supplemental, because it just says who did what, right?
Okay, yeah.
And then you can write that out in the method, like how many people actually had, you know what I mean? And then just,
Right, which I did, yeah.
yeah, and then put them in the supplemental figure. And then you can
Okay.
make the B bigger, BC bigger, right? So,
Right.
yeah,
So like these 4, just like this.
yeah.
Okay. And then everything else is straightforward. Oh yeah, so I wrote the cover letter for each cohort. Would you like to go over them right now, or I can print them out and show it to you in person?
No, I can read it right now.
Okay,
Perspective.
this is JC.
Oh yeah, go down. Yeah, that's fine.
Okay, and then this is the non-epithelial ovarian cancer, JAMA Oncology. One similar.
Okay, go down. Yeah, that's fine.
Right. This is Nature Communications for clear cell gynecologic cancers.
Go down.
Oh, yeah, this one. If you would like to provide any preferences for reviewers.
Names and contact information for qualified. And they may request the exclusion of certain referees. So how many do we have to suggest? It's mandatory.
Oh, they didn't specify. I don't think it's mandatory. I can,
If it's not mandatory, just leave it blank.
okay. Oh, should I even just take this sentence out?
We welcome the opportunity to engage with any qualified. What do you mean by engage?
Oh, I mean, I guess invite. I'm not sure. Because in the cover letter, it said here, authors are asked to provide. It just, it's just the one sentence that mentions this.
I'll ask to provide the names and contact information. And they may request exclusion. Are asked. Means like it feels mandatory. Huh. Let's submit and see what happens. And they, the worst thing that can happen is like, because usually when you submit in a blank box, there's a review suggestions. I haven't seen that happen in the cover letter, so just take it out and see what happens.
I'll just take it out. Okay.
Yeah. If the thyroid cancer paper gets rejected by JCO, the next one to go to is Nature Communications. To be,
Let me just write that down.
yeah.
Okay, and then this one is BAC. This is for JTO. It's not the short report because it's just way too short. Here. Oh yeah, I wanted to ask you. It says define abbreviations that are not standard in this field in a footnote. But like these BAC, MIA, these are standard in the field, right? And so I don't necessarily have to put these abbreviations in the footnote. Is that correct?
Correct.
Like CR, OS.
Right. Yeah, it's okay.
Okay. All right. I'll just ignore that.
Yeah.
And then this journal didn't require a cover letter. It just didn't mention anywhere. So I'm assuming I don't need to write a cover letter. Is that correct?
Correct.
Okay. And then these are the keywords. They only limit it to 3 to 5. Would you be okay with these?
Yes.
Okay.
So if
And
you
yeah.
actually go in and in the upload file upload, if you find cover letter category,
Yeah.
still write one and then just submit.
Oh, right. Okay. Yeah, that could happen. I'll just prepare something that's very similar to these.
Yeah.
This one is desmoid for JTO, JITs.
Okay, there's no specific individual that we requested. Okay, that's fine.
Leave it or take it out?
You can take it out. It's good. Yeah.
Or it says state if there are any specific individuals that you would not. Oh, okay. Oh, all right. Okay. Yeah, I'll just take it out.
If there are, because there are, so you don't have to even mention it.
Right. So for desmoid vulvar and small cell ovarian, these are all going to JITC. Would you still be okay with that?
Yeah. Yeah, it's okay.
Okay, that's all. And this is the signature I just found somewhere.
Yeah,
And lastly, this is the small cell ovarian. Take this part out.
yeah, it looks okay.
Great.
This, that was what journal?
That was JITC. The
Okay,
3 cohorts going into JITC.
okay. It would have been better if we submitted like, you know, with different time sequence. Yeah, that's, you know, my expectation was finish one, submit, next week submit, next week submit, you know, so that it doesn't look like we're submitting all at the same time.
For JITC or for other journals as well?
For everything, like it just says but then it's not, so I guess it's the same way, we get to Kurdrak's first blessing, and then we get to Patricia circulates to BMS. So it might be better to do that all together, because it's gonna be too many emails. If you do it this way, you could kind of attach all the journals, articles, and then just circulate at one. That's okay too, but you
It'll
know,
be in
Yeah,
multiple
it'll-
emails
yeah.
anyways, though, because the author list is different for each
oh,
cohort.
the author list is different, so it's gonna
Mm.
be all different. Yeah. Okay, so my suggestion is, even if it's by a day or two, just do it as you finish one. Don't wait till you get a whole batch and send it out. Like, it happened to, like, they won't have time to go through all, they'll feel overwhelmed. Like, I don't have time to go through all this, like even Kurdrak, so. So I know it's same thing, but don't feel like you wait till Tuesday. If you feel like you've done, like I said, once you're done, cover letter, just show it to me and send that out that day, and the next day you finish the other, show it to me and you'll send it out.
Mhm. Yeah, I wasn't necessarily waiting. It's just that I just got the risk tables for thyroid and I'm just showing you. This is like fresh off the email. And so I think I can get the thyroid ready. I'll send that out to the team today.
Sounds good. And
Yeah.
do you need other cohorts numbers for the,
Yeah, risk tables. Yeah.
so you
yes.
need, so work, you saw my email, so work with Megan.
Right. Yeah. I'm sorry about that. We did get the risk tables for other cohorts. It's just that I didn't get the full list. We're working on it.
Yeah, and Megan's pretty good, so Liam,
She is.
yeah, she should be.
Megan's been helping us like every day right now.
Yeah.
Yeah. And I'm sorry about that. Yeah, I got confused with the CMAC team, and yeah.
Oh, that's okay,
sorry
but
about
you
that.
sent her the clarification letter, right?
I did. I
Yeah.
did.
Okay. All right, very good. So that's the DART paper for now, right?
Yep.
Okay. And DART, it's been some, that the thyroid is poster at SITC, is that SITC? Right?
Yeah,
Yes.
that's Sissy.
The updated abstract, yes.
When do we know about SITC?
31st.
End of
This
July.
month?
Mhm.
Oh, okay. And when is the next meeting? It's AACR, right?
The next abstract deadline is AACR, yes.
Do you know when the deadline is?
Should be November.
November.
November. So Stas, let's aim for ITH something to, we'll work with Tempest Data and work on ITH, get something out for November deadline.
Okay, yeah,
Uh,
that seems reasonable.
Yeah.
So we
Dr.
also
Uh-huh,
We're with Picture Health.
uh-huh,
We've sent them our segmentations and the DICOM files for the initial 130.
uh-huh.
So they're working on it right now on their end, and we're working towards a deadline to submit abstracts to AACR together.
Picture Health.
Mhm.
Are they doing only radiomics or they're doing
They did
HN?
they did Pathomics too. We're still waiting for the path slides, so that's just going to take a little longer.
Okay,
Yeah, so a lot of the students helped me, Monica as well, collect the slide numbers. And so I just contacted Dr. Alexiev and those are slides from 111 patients, something like that. It's not the full 138 patients. Yeah, and he said he's very busy with clinical
yes,
stuff,
he's always
yeah,
saying
so
that, but it
yeah.
doesn't mean he'll not do it. It says he'll when he has free time, so,
Yeah,
but you were CCing me on all that, right?
yeah, yes. And I'll send him a thank you email after this meeting.
Yeah.
So
So
the-
what is
yeah.
the number of H&E slides then, total? I know Ether did the first batch, but the second batch.
Yeah, let me give you that number. Sorry, one sec. It's 55. It'll be roughly 160, 165.
Altogether,
Yes.
165 frontline immunotherapy slides.
About 40 of the patients didn't have path slides available at our institution. Is that what happened?
Oh, okay.
That was a question, actually.
Oh, yeah, yes. Something
About-
like that. There were many reasons, but yeah, most of it was like OSH or cytology only. Because we got rid of the cytology. Monica helped and collected a bunch of cytology patients, but yeah, we ended up excluding that because we obviously H&E slides couldn't be done.
Hmm. So you and Monica can do. So here's what you can do. Identify those ones that H&E slides are not with us. And we've done it with the adenoid cystic project. What you can do is you can reach out to the pathologist who read the H&E slide in that different institution. I would say the best way is to call the hospital. This is Dr. Hesong from Northwestern. I would like to talk to Dr. So in pathology department. If you don't want to do it, you can find that pathologist email. The best way to find it is through PubMed. If he published anything, you can find his email. Or LinkedIn, or wherever you can do some search to find his work email. If you can, the best way is to page that person, call the hospital. I even had a resident applicant or like lab applicant like page me like but talk to that doctor and say, hey, we have a retrospective protocol that looks at the H&E slides, the property analyzed by AI, predicting the response. Patient was treated in Northwestern, but somehow but we're not requesting slide. But if you have a scanner in your institution, if you can scan it and send to us, we'd like to acknowledge you in our paper and give you the results. That's what we can do. Some institutions say we don't have scanner, then you can say you could send us the slide to us that we can scan it, send it back to you. So we've done that with Seattle clinic. So why don't you sort of come up with that process, and you'll be surprised that some are willing to help us do our research And then you just make them know why this project is important, and then we will definitely acknowledge them in our paper, in the acknowledgement section. And then once the paper is out, we will send them our paper. Yeah, see, because like I think, getting few more patients that way makes sense, and that's what we exactly did for the adenoid cystic paper. Richard did the whole contacting process, but sometimes they have the scanner in place, and they just scan it, take a picture of it, and send us the JPG file. So it's doable. So Monica and Haesung, why don't you work on that to see if you can get access to that? Yeah, and then you can tell them this is our protocol and this is our IRB number. You can share that with them too, so that they know it's being done under the protocol. So let's work on that.
But it has to be a dedicated scanner. It can't be like on a smartphone photo.
Right. So the Lunit can tell you the minimum requirement of such scanner. But whatever it is, whatever scanner they have, we're not asking too much, because like, if you have an H&E scanner, slide scanner, you can send us the file. And then, just FYI, this is the minimum requirement. But they might not know the technical details, so just scan it on what you have and send it to us. The Lunit will let us know if this is qualified or not qualified. Okay, so let's do that.
Yes, if we find those, we'll be, so those are outside hospital patients are 29.
Okay.
So yeah, we might, if we get lucky, we'll get like 15 patients maybe additional.
Yeah. Okay, so that's the image. That's the picture health.
Mhm.
Picture
And
health.
then
Okay.
one more question, Dr. Chi. So for the radiomics project, we're working on the manuscript right now, but you know how like the analysis changed a lot from our previous abstract submissions. So I'm wondering if you wanted to do a new abstract submission potentially to AACR alongside the manuscripts, and maybe like embargo it if the timing works out.
Because a lot changed. So, well, I guess the one we submitted to SITC,
Mhm.
did it change a lot since then?
Because, and correct me if I'm wrong, Dr. Chian and Peter, but now we're doing it per lesion, not per patient.
So we are doing in different angles altogether. So we're gonna have to present per patient, per lesion, heterogeneity, mixed responders, or or universal responders. So we're looking at different angles, and these will all described in one paper. So the answer is yes. The one, the per lesion analysis, everything can be reported at SITC. And the abstract can be multiple. As Dr. Jian finds anything interesting, it should be per one idea, one abstract. So it could be multiple abstracts for the AACR, and then we can embargo it. Yeah.
Okay.
Yeah, I think this is very interesting project. So yeah, good point.
During your lab meeting, I just ran the SPSS, and when the tumor has what's that, the CDKN2A mutation and P10 mutation and STK11 mutation, then
Huh?
it shows a mixed response. I just found interesting.
Yeah, that's interesting. STK11 is well known to be immune resistant, along with KEAP1. So yeah, more mixed makes sense. Yeah. Okay.
And the very straight, it is correlated with the result of a very straight.
Oh, really? Okay.
Yeah.
So yeah, mixed versus so PR, PD, and how mixed, is in the middle of, is it in the middle or, in the three category. So that the mixed versus PD or PR, was that stable disease?
PD. Oh, I need to check. So
Yeah, okay. All right, Haesung, yeah, any other project updates?
No, I think I have enough on my plate.
Okay. All right. Okay. Yeah, Liam, give us update on other companies where we are.
So it's all with the company's legal teams. So I responded to some of the emails that you wanted to follow up on, which I did, and then our legal team got back to us saying that they're in the process of just meeting eye to eye with the company's legal teams. So it's just kind of been legal review back and forth at the moment.
Okay. What about the research companies? Where are we with Bertis?
Same situation. So we have for the biomarker studies, all of the agreements were sent to the companies, and like the companies are reviewing it right now. So cohort A was Bertis, OncoSite, OncoHost, and
Right.
one more.
Yeah, cohort A is really like Temple's there sitting, so I don't know why this is taking so long.
Yeah, it's just the legal process. So as soon as we get the agreement signed and in place, we can. I've already touched base with Rael. He's the PathCore
Yeah.
manager. So we're on the same page. As soon as we have the agreements in place, I plan to go there with Will to basically just like divvy up whatever samples we have left for the individual companies based on their requirements, and then have our PathCore ship it out to them.
Okay. Okay. What about apart from Picture Health is the one who are supporting our lab, who's supporting our lab, right? And then what about the other radiomics companies?
So Imagine AI, we have a meeting coming up with them. So we have an NDA with them, and they wanted to discuss more about kind of their stance and their platform, and then how we can collaborate on research projects going forward.
Okay.
So I think they wanted to kind of come up with like a few research questions together, and then start a research agreement that way. So I think it will be like the process will be kind of similar in terms of like them requiring the radiomics and pathomics and things like that. So we have all the data, so we can share it to them once we sign the agreements.
Okay. There's another company, right? Imagine.
So is it just Imagine AI, Picture Health, and then Image Trials? Image Trials we finished uploading.
Yeah. So let's do maybe let's get something maybe tomorrow. We could do tomorrow. Can we suggest maybe like, I know it's late for them, 7 to 7:30 in the morning. That's late for them, but if we do, I don't know, when's tomorrow?
Is this with Image Trials?
Yeah. Tomorrow, because I know Dr. Issa has testimony. So we'll have, it's 3 to 4, so. And it's, when is the very strat meeting? Biodesics?
Is tomorrow,
What time?
4 p.m.
4 p.m. Okay. Is it 4 to 5?
Mhm.
Okay. I mean, we can suggest to 5 to 5:30 image trial. Offer that we could do, like, we can ask, I don't know if Dr. Jian can come in 5 to 5:30. Are you picking up your children? 'Cause it,
Afternoon?
yeah, afternoon.
Okay, I'll try.
Because
Can access with phone.
yeah, your phone.
Mhm.
Say that will be 7 a.m. in
Mhm.
Korea. So we can offer tomorrow 7 to 7:30. In the morning, Chicago time. Or 5 to 5:30 in the afternoon, Chicago time. And see any of their company people can come and kind of give us an overview of what they are going to do and how we can work together. We want to just learn about the next step process. If we could do it tomorrow, then I can come in. If they can't, it would have to be same thing, Thursday, same thing, 7 a.m. to 7:30. Dr. Chian, you can still come in through Zoom from home, right?
Mhm.
So that offer that slot Wednesday, Thursday, 7 to 7:30 a.m. or yeah, 5 p.m. tomorrow. Yeah.
And I recommend the leader for this image trial project. Can Pegu do that?
Pegu, yeah, Pegu, you can be the lead. Pegu, you can be the lead, anything that's related to image trial.
Mhm.
But yeah, this would be radiomics project. So everything Pegu does, Monica, you have to be in charge because you're the
Okay.
radiomics lead. But anything that's pertaining to image trial, the um the details, the Pegu, you're in charge. Because they can, this can be very useful platform going forward. And anything you learn, learn from image trial people and the whole program, you're in charge of it. You can teach Dr. Yuri, okay, of what you can do with image trial program. You should be able to teach Dr. Yuri. Because that's his day-to-day job, doing resist and getting paid by doing resist. Okay, so that's that. And then there's an Israel company that does the H&E slides. What is that company?
Isn't that Imagine AI?
Oh, is that so? I can't remember the name of that company.
I believe Imagine AI is from like based in Israel.
Okay, that's the company that kind of predicts like ALK fusion from H&E slides.
I think so.
Okay,
Been a while, but that is what I recall.
okay. And the next is the JAX laboratory.
Mhm.
We should get, remember we were hoping to do a Zoom call with Chien, who's like the postdoc
Yes.
from her lab. So can you follow up on her? Because like what was the pending item? I think with the CDA was the.
Yeah, so CDA is still pending though. I'm just following up with our with like our admin team So we had emailed Christine. No.
Hmm.
So I had emailed. Oh, I'm forgetting her name. Kim Motter.
Uh-huh.
So we had initially emailed her all of them because she and Lizelle were working on them, and then Lizelle moved away, and then so it was Kim only, but then she was been MIA, so we had to kind of pick up the scraps and redirect all of them back to Christine Miller. And so
Mhm.
she has them now, and she's kind of redistributed these CDAs to some of her other colleagues. So we're in the process of like
Okay, so
recollecting the pieces and restarting the process.
okay, so let's follow up with her, CC me,
Mm.
ask her where we are. We need CDA in place between us and Jax. And once that's set up, we can yeah, and then just request that Zoom meeting with her team and whoever is, and Dr. Chion can join too if it's in the morning and night. And I just want you guys to learn the shareware, and Stats, this would be really good for you. This is like H&E slide, and forgot the program name. Do you remember? There's the most famous shareware for AI imaging analysis. And this is unbiased. It's not manual. There's nothing manual here. So we wanted to learn that and apply that to our H&E slides. So yeah, still not forget and just follow up and make
What is it called? OpenShare, shareware?
So the PI is Jeff Chuang, Jeffrey Chuang at Jack's Laboratory. He
Okay.
published a lot on like the TCGA H&E.
Oh, yeah, I remember you talking to him. Okay.
Yeah, but he
I'm back.
used this algorithm, and it's just open to everyone. So you should be able to use it for your project too.
Okay.
But what we agreed upon is was that his postdoc is GN was gonna teach us how to do it, and then we were gonna upload our own H&E slide to our their cloud, so we could do this together with Jack's team. Yeah, they've been very good. They are our kind of main collaborator. They are the one who did the neoantigen pipeline, remember? Victor.
Yeah.
So I think we can work on like H&E slide AI project with them as well. Okay, so let's follow up on that. And anything else, Liam?
No, that's it for now.
Okay. And Grace and Jessica, let's talk about what they've been doing and what they will be doing.
Right.
So yeah, you can.
Yeah, so far they've helped us collect data for the radiomics and the pathomics slide numbers, and that is all done. They've also helped us upload the image trials CD-ROMs, and then they've also helped us to kind of tie up the loose ends for the pacemakers projects, and they've also been helping us like re-edit some of the videos for the pacemakers and the GVM purposes. And they are also currently involved with doing helping finalize the variance classification projects too. And then they are eager to get involved with other projects now. So yeah, Grace and Jessica can chime in as well.
Yeah, just there are many other projects that we just want to figure out what will be the best way to get you guys involved in these projects. But I was talking to Liam yesterday, and I think getting projects published is a big challenge for our lab. So you could work on many projects that are being written up. Those are low-hanging fruits. And you can talk to Liam about what they are. And this is the great time to learn what projects are going on. And if some project sounds really, really interesting to you, you can reach out to me. You know, we're more than happy to have you be involved in any specific projects that really ignites your interest. Because this is the only time when you really learn about that what's going on who's doing what, not in their silo. Yeah, so tell me about that, but there are always projects that requires your help to I guess to take the lead on the project and really be in charge. I think you need to have clinical knowledge, MD or PhD level, and be able to mentor people. But as a student, any experience working with different data sets and writing papers together, these are all valuable experiences. So I don't think you should discriminate between projects. But my point is, if you really like one type of project, you can just say, I wanna have this as my main project and work on it. Yeah, so you could do that. But I think finding right patient and building a database and collecting biomarker data, these are really important, and these are also time consuming too. But this is where you learn and be able to learn from patients' database. So, you know, the things we talked about, making database for the first line therapy patients, second line and beyond patients, and neoadjuvant patients. I mean, you may not be able to identify the right patients, but be able to collect data for those patients who means like Tempest, Guardant, like 18 slide data, like, you know, what data is there, and survival data is easy, like when did patient die? Those are what patient students can do that too. So I think it's always good to be involved in that kind of data collection process. So Liam, make sure they're involved in those as well. Any other thoughts? Question, or just want to hear from Grace and Jessica, your kind of, you know, thoughts on what you've been doing and what you like to do. Grace?
Like what Dr. Liam said, I was just helping out with a lot of different projects and collecting data using Epic. And right now I'm just kind of updating pacemakers. But I think everything I'm doing, I'm learning something different. And I'm excited to help out with Sally and her variant classification, whatever she needs help with. And I think I'm also going to work on Tempest after the files get sorted out. But that's it.
Good. Yeah. I think the Tempest project, the ITH projects will have a lot of work to be done. So yeah, you feel free to talk to Haesung too about any help she needs with those projects. Same thing with Jessica. Jessica, any input from you?
Yeah, I've been pretty much doing the same things as Grace. And then tomorrow we're meeting with Sally to yeah, she's just gonna update us on like how we can help her. And then yeah, I'm excited to help with other things too.
Good. Take you.
Oh, sorry, Dr. Che, before we continue, Jessica, do you want to tell Dr. Che about this patient that you were interested in potentially interviewing for pacemakers?
Yeah, so she's a breast cancer survivor that goes to my church, and I wanted to interview her because so actually like last week I almost like wasn't able to interview her because apparently like her parents and her like in-laws
still like don't know that she had cancer.
Oh,
Yeah, so she had like second thoughts about that, like about like her interview being like public on YouTube and everything.
Uh-huh.
But like she said yes. Yeah, and like the reason why I want to interview her is
Uh-huh.
I just like talked to her briefly about like her treatment journey and everything, and she mentioned that when she had cancer, like she wanted to move to where her husband is working, but she decided to stay because of the church, like how like church members supported her so much. And like I also know that she started believing in like in her late 20s or something. So her testimony was about how like someone she knew from work and like her coworkers church like prayed for her
Oh.
to like be saved. And so I think in the interview I could also ask about that, and then like how her faith played a role in her faithful treatment journey.
So I guess faith-based organization, how that small group support can really help patients. Yeah, I think it makes sense. And then any question about faith has to be separate. Like I think yeah. So, but it has to be focused on the support and how that relationship helped her. And so why staying help, and then also the focus is also not support from her group, but also how her relationship with the care provider in in clinic helped her as well. So yeah, I'm more than happy to support that interview. Does is English primary, or yeah, Korean primary language for her?
She is fluent in English, but I was thinking doing the interview in Korean, if that's okay.
That's okay, but you have to put English caption below. Yeah, I think that's
Yeah,
fine.
sounds good. Thank you.
So same thing, it's we do want all the students be involved in at least one interview. So Liam has potential, so Grace during the summer and Will as well find a candidate. We do have someone, even like Grace, you can interview Dr. Iser. Because she hasn't done one yet. Yeah, so why don't you interview her? She's coming tomorrow to give testimony, so maybe you can ask her to give you like 10 minutes or something.
Is she coming in person?
That's what I thought.
Okay, I just don't know.
Yeah.
Okay.
Tell her to come in person if she didn't know. Yeah, because her testimony is from 3. She can come a little bit early to do the interview and maybe talk to Taekyu about the research projects. Yeah.
Okay.
Good. Yeah,
May
Taekyu.
I ask you some? Oh,
Oh yeah, go ahead.
well, I just wanted to show you something. Wait, did it go? Oh, this one, this is the RESIS PFS and OS of the translational cohort. We didn't get the risk table for these.
Uh-huh.
Should I ask them for the risk tables, or do you just want it out? I
It's-
just wasn't sure how important this is.
It's supplemental. I think it's fine. I think unless the journal asks, it's fine to go without. But at the same time,
Mhm.
so is this a requirement of JCO?
Yes.
They say every Kaplan-Meier curve should have. Show me that language. All Kaplan-Meier plots must include risk tables. So I think they'll be okay with supplemental figures. However, I don't want to bother Jen too much with multiple communications. So let's just ask. Just say JCO requires all plots must include risk table. So have her generate one for the translational cohort.
Okay.
Better to
That's
be-
good that you posted that. Now I need to add risk tables to mine.
Yes. It's always good to have one, but sometimes you don't, but need it, but yeah. Okay, good. So take your update from you.
Yes. Okay. First of all, I understand that you are concerned about the VeriStrat project, and I was working on segmenting last week's to make the results for radiomics ASAP. So I'm sure I can put the VeriStrat project to priority this week. And as I told you yesterday, I was working on rechecking the data by myself about the PFS and OS data, and I told you that some data was inaccurate, and I would like to elaborate. Yeah, I found out that many people were involved in this project and collecting the data, and let's say that when one patient showed progression, someone defined the data progression as the date when the follow-up CT scan was taken, but the other defined the data progression as the date when a physician wrote a clinical note that it is actually a PD. So I guess I have to go over all the data, that all the data was collected with single criteria. So, okay, and this is for VeriStrat. And for the clinical proposal, I am afraid that the statistician you told me to contact named Dr. Alfred Redbaker didn't reply. So I ran multiple algorithm by myself, and I said 0.1 for alpha value, 0.1, and 0.66 for hazard ratio, and 80% power, and total 236 patients enrolled. And are these values okay for you?
Do you have a screen you want to share or?
Okay, I'll share. I'll send you my the document link, and I'll share you. Okay. This is my document link, and I'll share you my screen for you. Wait a second. Okay. Is there any part you want to look into?
So tell me what's your question?
Okay, yeah, I ran a multiple algorithm, and yeah, here's I said hazard ratio 0.66, and for alpha 0.1 and 80% power, and there are 236 patients enrolled for that.
Okay, go down. First of all, we don't do 66. It's either so hazard ratio of either 0.7 or 0.75 or 0.8. I think hazard
Okay.
ratio of 0.8 may be okay, because the smaller the gap, the lower the sample size.
Okay. Mhm.
I would just Yeah, try all three numbers and tell me what the sample size is, and you're doing 1-to-1 randomization.
Yes.
Yeah, and just show it to me what those numbers are. Yeah.
Right now.
No, after this meeting.
Oh, okay.
But look at other phase 2 trials to see what power and alpha they used, and we just don't want to be outliers. Okay.
Okay. Most of the clinical trials like Nivo chemo and Pembro chemo, I checked all that as like about 80% power to like 0.05 alpha and 0.7 at like 0.7 hazard ratio. So I tried to set alpha to around 0.05, but the sample size went too big. So
Right.
yes.
What about why did you choose 0.66?
To like yeah, I didn't know that this should be like 0.7, so I tried to set the number of patients in between 200 to 250, so that's the reason.
Yeah, I think 0.7 is okay. But let's see what happens with 0.75 or 0.8, and yeah.
Okay.
So you said that the statistician is not available?
I believe so. He's not replying. I've sent him an email, and yesterday morning, the earliest morning possible so that he can check the email, but there's no reply. So
Okay.
yes.
I will ask some other people. Yeah. Who's our cancer center statistician is now. Okay. Next.
And I'm afraid I asked you about this yesterday, and I want to be sure that the overall study duration should be like 24 to 2 years, and so the expected time for patient treatment is like normally a neoadjuvant treatment takes 9 weeks to 12 weeks, so I said it 3 months. So the follow-up duration should be 21 months, right?
Why is it 21 months?
Yeah, I just guessed that the overall study duration should be 24 months, so I just subtracted 3 from 24. I don't know if this is right.
Oh, no,
like,
no, that's fine. Expected time for patient treatment is just you can put 6 months.
6 months,
Follow-up duration, you can put 24 months.
24 months like this. Okay.
Yeah, it's okay.
And I made a schema for this. I'll just can you see this PowerPoint picture?
Yeah.
So I made a schema, so I wanted to check I made it right.
Yeah, looks fine.
Okay, thank you. And all the rest looks fine for me. So like for this part, data management plan, I just wrote some data that we need to collect, like gender, age of diagnosis, or something like this. So is it okay for me to put this information like this, like so on?
So this is more, yeah, just talk to Liam. It's more about, we will de-identify and store into a password protected. There's a language we use for protocols like this, and even in the DART study or other IITs, there's some language about that. So maybe Liam, you can help kind of give him more standard language. Now it's more about how do you store and analyze in de-identified and safe place, and yeah.
So, Dr. Che, this is both the proposal we're sending to Novocure and also going to be like the draft for Northwestern IRB approval, right? Like the protocol.
Yeah. But Novocure has to say we're gonna fund this. So this is the workflow is this, and Haesung, this is the same thing for you. So how do we do clinical trial? So this 101, how do you open a clinical trial and you only have an idea? First is proposal like this. You just write up a proposal. Hey, so this proposal is saying we give chemoimmuno and then do surgery. Can we do Novocure? For those of you who do not know, this is a tumor treating field. Do you know how magnetic field can, so if cancer cells divide and then there's a spindle cell, and when cancer cells divide, if you have magnet right next to cancer cell, and then you move magnet like this, and then like this, constant, and then it's going to confuse cancer cells, it's not going to divide because of the interference caused by magnetic field changing. Because how that lines up is affected by the magnetic field. So it's like having magnet right in front of your chest and the back of your chest. So you wear a vest that has a magnetic field in it by electricity changing here and there. So you just wear a vest and that's going to be enough to stop cancer growth. So this is really thinking outside the box, right? We think about radiation, chemo, immunotherapy. None of that applies here. It's the magnetic field and it's not radiation. So there's no harm there. Because X-ray is harmful. That's why how Marie Curie died. So the idea is, can we wear that vest with chemoimmunotherapy before surgery? And Dr. Jian, we can do that for your breast cancer patients too. Very simple idea. And can that increase PCR, event-free survival? I asked the company, no one's doing this. And I say, why not? We can quickly write up a proposal and get the grant to run this trial.
So Dr. Chi, if this is funded, are we going to run this through the CTO or are you going to do it internally at our lab?
No, this is clinical trial, so it's CTO. So proposal first.
Mhm.
If they say we like the idea, we're gonna have Hugh Anderson, right? Is that Hugh Anderson?
Hugh's the finance person, yes.
Yeah. So we say this is IIT, and then this is the sample size, and this is the intervention. This is the blood draw we want. We might. I don't think we need to do EKG or urinalysis. We don't need anything. We just need study coordinator and data manager. Everything is standard care. And we just need someone to, provide, be in charge of getting the device and provide, and educating how, how to use that device and all that. So that will be more like, how do we budget things? So we get the budget and then we get company approval for the budget. Then we write up the protocol. We go to SRC, IRB, and then we get the budget approved. And then we launch the trial. And we're gonna have to see if we want to do this because if it ends up being 100 patient, 150 patients, I don't think we have enough patient for that. So it would have to be 2 or 3 center trial. That means a whole lot of different things for budget purpose. Because every institution have a different budget. So we'll worry about it when we get there. So first is, do you like the idea? And they say no. I think they'll turn it down. If they like the idea, then we go to Hugh, get the budget in, and then submit again. If they're okay with the budget, then we'll write up the whole protocol, submit to SRC, and then IRB. Yeah, so that's how we do the trial. And same thing for you, Haesung. But this is what you do as a faculty. This is more like a faculty level job.
Right. Dr. Tegel, was there a space where they were asking for an actual like specific budget, like dollar amount?
Yes, as they require like
Right.
the estimate of the budget required. So
Okay. So initially we're just going to submit the proposal to them, and if they're okay with the concept, we'll get the budget together and then get the budget approved as a secondary, as a second step.
Right.
Okay.
Because it takes time. I don't
Yeah.
want to give Hugh work when there's
Yeah, no interest.
yeah. All right, any other questions, Tegyu?
that's pretty much it. Thank you.
So Dr. Chief, for this study though, what are we going to have Novocure pay for? So they're going to pay for the data manager, the coordinator, the device, obviously. Are we going to have
Everything. We're not paying for anything. We're having them pay for everything. IRB fees, SRC fees, everything.
Okay, but any like blood draws and anything like that, or will like the clinical aspect all be like standard of care and insurance coverage?
Mm-hmm.
Okay.
Yeah, I mean, you guys everyone in this meeting can feel free to think about translational work, tissue and blood. Is there anything interesting from this trial? Because I don't know enough about the magnetic field biology. Like, what are we looking at in terms of, PK, PD type of, what biology should we look into to prove, for the proof of concept? It's really hard. Like, let's say if a drug is not working, we look at different pathways and what is the pathway of resistance. Like here, I don't know. I don't even know what I should be looking at. So I don't know if, if I do blood work, like what are we looking at, right? So right now it's just pure clinical. But you're welcome to read into their literature. If you go to Novocure homepage, you'll learn about their science, biology. They have a beautiful YouTube video explaining everything. So feel free to learn and think about new ideas and pitch it to me. I'm more than happy to think about anything that can be added into this.
So as of right now, we don't want to necessarily have these patients overlap with either the biomarker study or even the SAINT study, right?
I mean, those studies can be applied like at the same time.
Okay.
Yeah, two separate consents.
2 or even 3, depending.
Yeah,
Okay.
it's still fine. Yeah. Okay, anything else take you?
That's pretty much it. Thank you.
All right. Anything else? Any other questions or comments before we wrap up?
Yes, I would love it if you could look over the introduction that I
Oh,
wrote.
yeah. Yeah,
Um, so yeah, Dr. Jion's been teaching me how to write a manuscript. And so I'm sure there's plenty of mistakes and corrections that you can point out for me. So is it being shared with you guys?
yeah.
Okay.
Actually, can you send me the link? Because it's better that I drive it.
Yes, I'll send you the link. This should be sent to you. Messenger. Did you want me to share screen still?
Send it to the chat
Ah.
box in the Zoom.
Okay.
Okay. got it. Predict tumor response and biomarker status. Is this the title?
That was a previous title, but I think we have to revise according to the research that Professor Uri will give you.
Yeah, because we're gonna combine pneumonitis here too, right?
Yes.
So depending on what we find, I feel like response and pneumonitis are two different things. You can make it into a two different paper too. Because we're talking about response here.
Mhm.
I don't think Peter talked anything about pneumonitis here. Right?
If the research from the pneumonitis is strong, we can make a definitely different paper. But if it is weak, then I would just put as one of the table or like
Yeah.
that.
Yeah,
So,
I like that. Let's see what we find first and then decide. But because biology is so different, I like
yeah,
it.
yeah, I agree. Yeah.
Because we're talking about the therapy.
Mhm.
What are these numbers?
Oh, I forgot to remove them. Those are the PM IDs.
So it's not to predict changes in intrathoracic lesions.
Well, for the purpose part, we are still wondering. So
Yeah. So the resist has been done outside in thorax too, correct? So when you say you are predicting intrathoracic lesion response, you are ignoring the extrathoracic response when you say that. So we have to
I
be
got it.
careful. You have to be,
Mhm.
so using intrathoracic lesion radiomic features
Mhm.
to predict the overall response,
Okay.
okay, is the goal. And under that subheading, intrathoracic lesion heterogeneity, does that play a role? And does the intrathoracic lesion features predict intrathoracic lesion response better? Or overall response better, or extra a thoracic lesion response better? So compare all of that. And in the intrathoracic lesion, is there heterogeneity in terms of response? You can look at the waterfall plot and see, what the heterogeneity is there. And between intrathoracic and extrathoracic, is there heterogeneity in response there as well? So, and then does the heterogeneity in terms of, if you treat all the lesions same, so let's say if you treat intrathoracic, extrathoracic lesions same, if you see heterogeneity is all lesions per lesion, heterogeneity in response in terms of mixed response, how does that affect overall survival? And heterogeneity in response in intrathoracic lesions, does that alone it relates to overall survival difference? Or is it between the intra versus extrathoracic lesion in terms of response? And you can, for that analysis, you can combine intrathoracic and extrathoracic lesions together and see the response rate difference there. And then how is that heterogeneity intrathoracic versus extrathoracic response difference predict response, predict survival in terms of PFS and OS altogether? So there's a lot of questions we're asking here, and I want to be very clear cut on all these questions. There could be 20, 30 questions, and you can learn all, learn about all these questions, how they're different, how the analysis is different. But at the end of the day, the number one question is, because of what we did, this is an inherent limitation, but it could be our strength. We've only analyzed radiomic features of intrathoracic lesions, as opposed to all the other radiomics papers that looked at liver lesions and brain lesions, everything. For whatever reason, we only looked at intrathoracic lesions. So by utilizing intrathoracic lesion features, can we predict the overall survival outcome, overall lesion response? Okay. If the answer could be yes or no, and then we can go on to ask, does intrathoracic lesion properties or radiomics features particularly predict intrathoracic response better versus extrathoracic response? Because we're doing per lesion analysis, we can ask this question. So all of these are very important, So come up with a title that will encompass all these thoughts. So the heterogeneity, so the number one question is still intrathoracic. Can utilizing intrathoracic radiomic features, lesion radiomic features, predict patient outcome? And how does heterogeneity in heterogeneity in per lesion response predict overall survival outcome in patients who are all treated with immunotherapy containing regimen? So these are the two questions. One, intrathoracic, be able to predict overall, and heterogeneity in response correlating with survival outcome. So those are the key features. That's why having pneumonitis is another, like, what do you guys like? Why is there so many different topics? So let's say if the heterogeneity question yields too many papers and graphs, that's another paper. Heterogeneity in response. I feel like that it's a really big question we're asking. But then intrathoracic lesion property alone at patient level as well as per lesion level, how can it predict a response at per lesion level and at patient level, PFS-OS? And just using intrathoracic lesion level, is that good enough or it's not good enough? So we have two different questions, okay? And both have not been asked in conventional radiomics paper. So that's why both are novel.
Mhm.
And that's why I prefer having pneumonitis as another paper, because it's like
Okay.
you added, what are you guys trying to talk about,
Is
So-
there any answer regarding about the questions without the radio mix?
Without the radiomics, I think without radiomics, there's been papers out looking at per lesion, how per lesion response is so important in predicting overall survival, like just RECIST wise. So you should find that paper, because there's gotta be a paper somewhere. But that concept is so important. And we did oral presentation at, cite this oral presentation or abstract by Megan from SWOG. And Liam, you can help us find that oral presentation by Megan. Megan's the first author, and it was just the past ASCO. She did oral presentation with the DART patients. So all DART patients, when she looked at response rate and then PFS and OS, there was a linear relationship. So the 30%, 20% is not a cutoff. So that response rate at linear level predicted patient outcome really well. So I think the whole point is the RECIST to 20-30% is really just random or arbitrary cutoff. And there is no like threshold type of justification. It's all linear. And that's why maybe looking at per lesion may be more meaningful. And the heterogeneity in response prediction will have, because if same agent shows mixed response, that tells us that tumors are really heterogeneous here. There's a lot of metastatic sites that are heterogeneous, and that means much. And this goes back to Stas's ITH projects. And ITH means that whether you get targeted immuno or chemo, you're not going to do well. So I think just looking at the per lesion mixed response pattern, you'll be able to just whatever therapy patient gets, and then maybe and then, I think in the future, they will do worse. But let's say the more combination therapy they get versus, just like breast cancer world, they get single agent chemo. So more combinatorial approach versus monotherapy approach, there may be a differential outcome there. So I think the heterogeneity question itself begs for like one big question. And then the other is intrathoracic is enough to predict per lesion or per overall response is another idea. And then the toxicity is another idea. So, and then that toxicity is important because you don't need to know what it looks like in liver, like intrathoracic trait, because we're only looking at pneumonitis. So that might be a very a whole different concept. So the more I think about it, it begs for 3 different papers.
Okay, what if we make paper regarding mixed response with the 209 patients, because we have already all resisted data? Because I'm trying to distinguish intrathoracic and extrathoracic from 131 patients. But obviously 130 patients, 31 patients, they had all intrathoracic lesions, but only 20 to 30 patients had extrathoracic lesions.
Good point.
So yeah, but that is because we excluded a lot. The patients only had extrathoracic lesions. So when we go back to 209, of course we can see that answers from
Yeah,
the
so
question.
good point. So for the second heterogeneity or mixed response paper, we don't need radiomics for that. We don't need radiomics.
Okay. Yeah.
Yeah.
Who's gonna be the leader for that?
Oh my.
Okay. But I think it's because the problem is the Professor Yuri is not a physician. It is quite difficult to understand all these complicated concepts. So it's my thought, radiomic paper should go to be a little bit simple. And then we can see the mixed response with the 209 patients.
So here, yeah, because like it's hard to. So radiomics contribution in the mixed paper.
Oh, there's nothing regarding about that.
Yeah, because we want to be simple. Like radiomics able to predict mixed response is too much because you have responder, non-responder training is good enough. I mean, if you want to train for mixed response, I mean, we don't have enough sample to have 3 category training. So
yes,
in that regard, radiomics has, yeah, we could be simple enough. I agree. I think mixed response could be a pure clinical paper.
Oh, yes, of course.
Yeah, pure clinical paper.
Yeah, I saw many gene mutations was significantly correlated with the mixed response.
Yeah. This is very clinical, and I think Dr. Jian, you understand all the clinical aspects.
Should I change my major?
I think you're trained as medical oncologist.
Let's skip surgeon.
So I think Haesung's doing a lot of things. So I think Minjung would be a good person to take over this. But she's coming late in the fall. But until then, I think you can kind of mentor, the people you're working with.
With Peter.
Yeah.
Peter. Yeah.
Peter and Taegu.
Yeah.
He's gonna be here. But like, but it has to be MD level, like understand.
Oh, yes. Mhm, mhm,
treatment.
Sure. Yeah.
Has to be MD. It's very clinical.
Mhm.
So work with Peter and Taegyu and keep writing it until Minjung comes and she can take over and submit. If it hasn't been done by then. And then for the radiomics project, the intrathoracic lesion predict overall is a separate project. Let's just leave it that. And then you could mentor, same thing, Monica, Peter, and Liam's, you can help, and whoever comes next can help out. But I think you're the one who sort of understands the ins and out, and then I believe you'll come to at least, you know, a few minutes for the lab meeting. So you should be able to just mentor them for the two projects, and let's see what the pneumonitis shows and decide for that.
Yes.
But the one who will actually take over, if not done by then, will be Dr. Minjung, I think. Yeah, let's just done by then. And Haesung can always help. I just feel like she has a lot of projects, so she could always help out. But like, I don't want her to be the lead. There's too much on her plate.
I think once I'm done with the dark papers, I'll start seeing what else I can do. Because
Yeah,
the
yeah,
TAP
yeah.
project, the Tempest stuff is kind of like it's just going very slowly. So in the meantime, I can work on something else.
Yeah. You can help out with it. Yeah. Okay. Good question. Anything else, Peter?
Oh yeah, and one more thing. This is not, I don't think it's coincidence. So this Sunday I tuned into my church's online service, and I didn't know that one of our lay elders at my church is actually a gynecology surgeon. And so he gave a really, really convicting sermon, and I was wondering if I can interview him for the Pacemakers project when I go back.
Oh, he's in Austin?
Yeah, he's in Austin, and he's one of the elders at my church, and yeah, he's pretty similar to you when he preaches. He talks a lot about suffering and rejoicing in the midst of it, and it was actually a really good sermon. And so I feel like it'd be fun to interview him.
Okay. So same thing with Jessica, Peter.
Mhm.
Yeah, definitely we welcome any interview. And if he's practicing in US, you can interview definitely in English. Well, why is he Korean speaking or English speaking?
No, he's actually Hispanic. His name is Devin
Hispanic.
Garza.
Oh, okay. Yeah, definitely English. So separate faith questions from patient-doctor relationship question, and the pacemakers will be all about, you know, support group, patient, caregiver, team relationship, as well as the whole journey itself, how, you know, what are the lessons they learned and all that. Because I know the previous interviews has been more focused on like, what was your training? Like, what is your research? You know, that's not the focus. Those can be icebreakers, but just understand, you know, why we're doing what we're doing. And then if they have any testimony about their faith, you could kind of record it in a different setting. We're happy to make that video into more like a faith-based video. Same thing with Jessica. We want to kind of separate those questions and record it differently. But I don't want too much faith story, because everyone will have different religions, and I don't want that to be like in the way of like the pacemakers movement. So it will be you know, you guys can do interview, but then those questions can be edited in a separate media. All right, very good. You could do that. Anything else?
No, that's all for me.
All right, very good. So we'll see you guys next week in the lab meeting, including Dr. Jian.
I will be in flight.
Oh, you're
Yes.
flying. Okay. then
Yeah.
follow.
I can't access. But then the next week, we'll see.
Yeah, we'll see. Okay. All right. We'll see you guys. Thank you.
Thank you, Dr. Shay.
You.
Thank you.